Dr. Christophe Conrad, Scientist, Unit Major Policy Issues, International Relations at Powell Ehrlich Institute. Professor Dr. Duncan Craig, Director and Chairman in Drug Delivery at UCL School of Pharmacy. Dr. Gabriel Dahlman, Founding Partner and Principal Consultant at Biopharma Excellence. Zubair Kumar Pasak, Senior Industry Specialist, IFC, a member of the World Bank. Dr. Wieland Wolf, Chief Executive Officer, ProBiogen.
Good morning, doctors. We'd like to welcome you all in the first national conference, Harnessing Biotechnology to Improve Healthcare in Egypt. Thank you all for coming. We'd like to announce the start of the conference now with a welcome address from Professor Dr. Tamer.
Thank you very much for all dear guests and honorable attendee. It's my pleasure to be here between you. It's my pleasure to be here with you, and I'm very honored to be under the auspices of Her Excellency Dr. Hala Zaid, the Minister of Health and Population in Egypt, to represent the Ministry of Health and Population, and also to represent the new coming star Egyptian Drug Authority. So we are here to speak about our first national conference talking about harnessing biotechnology. And in this, I would like to start a very, very quick presentation just to welcome a ceremonial way with the quorum attending this conference with scientists from Egypt in order to make the pillars for engaging the academia and industry to gain what we are expecting from biotechnology in Egypt. On behalf of Minister of Health in Egypt and Her Excellency Dr. Hala Zaid, we welcome you all. And also, we need to announce that we started our new Egyptian Drug Authority that will take over all related subject for regulating pharmaceutical, biological products, medical devices, and it will replace very soon, inshallah. Also, I'd like to welcome our colleagues from Paul Ehrlich, that we will be happy to have a very strong cooperation and joint venture for the coming few years, partnership, twinningship, any kind of cooperation we are looking to. So we welcome them to Egypt, and we would like to wish them a very pleasant stay in Egypt and a very fruitful discussion with our team. They started yesterday, today, and tomorrow, inshallah. Thanks for Mina Farm, ProBiogen, and all our partners to organize this esteemed conference, and also for New Giza University. They will be the academic part or arm for this conference, inshallah. We do have very strong will and directions from His Excellency President Abdel Fattah al-Sisi that we have to make us ready for the coming era. And coming era, we are expecting to start by 2025, where biological and biosimilar products will replace greatly all conventional generic products and medicines. That's why I was so lucky. I started my PhD studies in Lund University, where I have learned what does it mean biotechnology. And I got the specialty for biotechnology from Lund University, Sweden, and since then I started to work with biotechnology. They have changed a lot of my thinking and thoughts. These now are my dreams, how we can be organized. When we are talking about biological markets, we can see very dramatic change in the market and figures. We are talking about industry that has an income now up to more than 200 billion US dollars, and the forecast by 2025 it could reach up to 400 billion US dollar. This is a great amount, and Egypt has to be ready, as we have done in 1960s, where we started the generic pharmaceutical production and the industry. That's now why we are making gathering and strong cooperation with private sectors to initiate and establish the biological and biosimilar industry in Egypt. We are very aware about the competition between biological products and biosimilars, but actually when we are looking for the pharma emerging markets all over the world by statistical numbers and figures, we can see that biosimilars also are coming very strong to the market. That's why in Egypt we are adopting a strategy to keep the balance between having the biological products and biosimilars on the same foot. We do care about biological products innovation. Also, we do care about biosimilar industries in Egypt. We are here to announce that we are starting our initiatives, we are starting our strategy to adopt biological and biosimilar industries in Egypt, and we will be ready by 2025 to be among the main pillars in the world for this huge industry, inshallah. Also, we are announcing that we are hand in hand with the private sector in order to establish this very strong industry. Welcome you to Egypt. I wish you a very pleasant conference, fruitful discussion. Thank you for all my colleagues, and thank you.
Thanks a lot, Professor Dr. Tamer. And now the welcome address for Professor Dr. Ahmed Sameh Farid. Please join me in welcoming him.
Thank you, and good morning, ladies and gentlemen. It gives me great pleasure today to be among such distinguished guests and speakers. I would like to thank you all for coming and to give us from your time to share your knowledge with us today. We believe it is very important for the years to come to share knowledge between the healthcare providers. And when I say healthcare providers, I always remember when we have started New Giza University and we said we will start with the medical division. The School of Pharmacy did object. They said we are not medical university, we are the health and life sciences, we are health sciences and pharmacists as well in the healthcare system as equal as any other doctors or dentists or whatever it is. So we did change it to life and health sciences, and we believe today that the School of Pharmacy is the main pillar in our university, and we believe as well that in the coming future, the School of Pharmacy will have the lead for innovation and creativity to find more and more products that will help the medical profession to achieve its goal. We have different aspects in our healthcare system. We have the medical providers or the healthcare providers. We have a very strong R&D impact because R&D in the future is the future of the profession. And we have the industry which will support us. Eventually, in this part of the world, the industry contacts and believe in Egyptian R&D was limited. But I think in the last few years, especially with Mina Farm coming into, we are developing some sort of strong and true partnership in R&D with the industry. I think the future is very challenging. We always ask ourselves and our academic partners, UCL, are we offering our students in our university the kind of knowledge that it will help them 10 years from now, or 10 years of now, science will change. The knowledge will change. The medical treatment will change. The biosimilars will be there. The genetic engineering will make the gene transformation, and it will make the gene therapy different. And I always remember when we started 10 years ago to think about the School of Medicine in Egypt, we had a partner from the States, one of the top 10, and they offered us a curriculum called Gene to Society. And when we said Gene to Society, what is Gene to Society? He said the future is genetics, molecular biology, all of this. All what we have been taught in the medical school years before will be obsolete. So the future is challenging. And I think the future is in the hands of the R&D, in the hands of the partnership with the industry, in the hands of the loyal scientists, especially to their countries, because most of the R&D we get from the other world, not from our world here in Africa and Egypt. We need more impact for our R&D in our country, especially for our national health care problems, national health problems. So it is a big challenge. So I believe that such a meeting is a challenge for all of you to think with our honorable guests coming to share their knowledge with us, to discuss with the industry how we can make the future different, how we can be of help to the coming generations, because definitely the kind of knowledge that we were taught 30 or 40 years ago is not valid anymore in the coming years. I would like to thank you all. I would like to thank Mina Faram. I would like to thank our distinguished speakers who came. I would like to thank the Ministry of Health for being there with us. And I wish you all a successful meeting, and thank you.
Bismillah ar-Rahman ar-Rahim. Good morning. It's a pleasure to me to be a chairperson for first session in this very important conference. This is the first conference for biotechnology, which is a partnership between Ministry of Health, CABA, NORCP is the body which is responsible for biological regulatory aspects and regulations. So that's very important in Egypt to initiate this kind of conference to integrate the work between the partnership to build on that future plan to 2025 to be a leading country in the Middle East in the biological industry. And it's a pleasure to me to introduce Professor Dr. Mahmoud to start the first talk in this conference, and he is the chairman of the conference, and I thank you him for all the effort beyond this session that happened to give us this chance to be here now. Thank you, Dr. Mahmoud, and you have to start.
بسم الله الرحمن الرحيم. Starting with welcome for all partnerships in this conference, and I'm very happy to find many of my professors participate in this conference, and especially Dr. Ayman Khatib. He is first really happy and will be, and many of professors that participate also they have a rule after God to be here. So I'm very thankful for all of them, and also for many biopharmaceuticals, and she made a lot of work to establish or manage this conference, and also to be collaborate with Paul Ehrlich. I'm very happy to do that, and I hope that my presentation will be starting for the conference to give some information about biopharmaceuticals and biosimilars. So, starting with the title is Biosimilar Developments and Economic Implications. Starting for biopharmaceutical, this is biological, pharmaceutical product manufactured in, extracted from, or semi-synthesized from biological sources. They are produced from living system by biotechnology and used for therapy, protective purpose, or for in vivo diagnosis. The first biopharmaceutical approved by FDA in 1982 is the recombinant human insulin, followed by the first recombinant vaccine, which is recombinant hepatitis B vaccine, and also the first anti-cancer recombinant interferon in 1986. After that, several biopharmaceuticals were developed, and they are under different classes or families, as shown in the list. As compared to conventional drug, biopharmaceutical molecule is larger in size, complex in structure. For the ingredient, its complex ingredient also, manufacture processing is a specific and complex biological process. Finally, regarding the cost, it's higher in cost as compared to the conventional drugs. Transitioning from traditional pharmaceutical to biopharmaceuticals, WHO states that access to essential medicine is part of the right to health. Partners of death are changing. Partners of diseases are changing, with non-communicable disease, which are being responsible for about 70% of all death worldwide. Innovation in medicine is required. Better treatment means more effective medicine for more diseases. Biologic drugs are transforming treatment of other hard to treat diseases beyond cancer, including asthma, MS, heart diseases, inflammatory bowel diseases, because these targeted therapies offer improved effectiveness compared with past generation of small molecule medicines. Biopharmaceuticals represent the fastest growing sector of pharmaceutical industry, with all current indication suggesting double-digit growth annually over the next decade. The growth is mainly due to greater efficacy and reduced side effect of biopharmaceuticals. As shown in the figure here, which is the worldwide pharmaceutical product sales, biopharmaceutical share in the percentage of prescription and OTC sales, starting with 18% in 2010, rising up to 28% in 2018, and suggested to reach to 32% in 2024. Worldwide pharmaceutical product sales from top 100 products, biopharmaceutical sales, it's about 88 billions in 2010, rising up to 162 billions in 2018, and suggested to reach to $178 billion in 2024, which approximately 50% share from the total product sales of the top 100 products. The global pharmaceutical market was valued at 237 billion in 2018 and estimated to be valued at 389 billion in 2024, registering a compound annual growth rate of 8.59% compound annual growth rate. Regarding the top biopharmaceutical product sales worldwide in 2018, the product sale range between $5 billion to $20 billion. Biopharmaceutical high costs have an impact on access to drugs. Evidence suggested that there are serious gaps in the availability of biological medicine in many countries all over the world. One potential route for saving cost of therapy is the use of biosimilar medicine to replace originator medicines. Between 2018 and 2024, it's predicted that 251 billion of currently patent-protected sales could be replaced by more affordable follow-on versions. Biosimilars. Biosimilars is a biological medicine highly similar to another already approved biological medicines. They are approved according to the same standard of pharmaceutical quality, safety, and efficacy that apply to all biological medicines. Biosimilar can be manufactured when original product's patents expired. Regarding regulatory approval pathway, as compared to the reference medicine developments, biosimilar developments require more analytical and preclinical studies with few clinical trials and clinical pharmacology studies. Compared to the reference product, biosimilars may create opportunity to lower spending for payers, employers, governments, and patients. Two factors are primarily drive these savings. The first factor, developing a biosimilar costs less than a reference biologic. Biosimilar can expected to take 8 to 10 years to develop at a cost between 100 to 200 millions of dollars, compared to an estimate of $2.6 billion for developing a new drug or biologics. As a result, manufacturers have fewer expenses to recoup, which allows for biosimilar to have lower list prices. The second factor, biosimilars contribute to competition in the healthcare system. As the number of treatment choices increase for a particular disease or condition, manufacturers may be motivated to lower the prices of their product to maintain or increase market share. In addition, manufacturers of reference products may also discount their product. This price competition increases savings. Impact of biosimilars in Egyptian market. Just to give some examples of biopharmaceuticals in the Egyptian market and their prices or sales along different years. Trastuzumab is a humanized monoclonal antibody used for treatment of breast and cancer and gastric cancer disease. Rituximab is a chimeric monoclonal antibody used for therapy of non-Hodgkin lymphoma. Starting from 2015, the product sale is about 229 billion Egyptian pound and reaching to 480.8 million Egyptian pound in 2018 with compound annual growth rate of 20.36% and average cost per year 353.5 million Egyptian pound. Rituximab the product sales starting in 2015 by 163.5 million Egyptian pound and reaching to 282.4 million Egyptian pound in 2018 with compound average growth rate of 19.98% and average cost per year 242 million Egyptian pound. If we take this information in consideration, more than 123 million Egyptian pound can be saved per year, when currently trastuzumab can be replaced by more affordable follow-on version, and more than 84 million Egyptian pound can be saved when currently rituximab sale replaced by more affordable follow-on version. Other examples, anti-tumor necrotic factors antibodies such as infliximab and adalimumab. Both of them are monoclonal antibody present in the Egyptian market, and the same, there is a rise in the product sale along different years from 2015 to 2018, and with average cost per year 41 million Egyptian pound. And also adalimumab between 2015 to 2017, rising up in the product sale with an average per year about 60.5 million Egyptian pound. Also, we can expect that more than 14 million Egyptian pound can be saved per year when currently infliximab sale product replaced by more affordable follow-on version, and more than 21 million Egyptian pound can be saved per year when currently adalimumab sale replaced by more affordable follow-on version. These are some of the biosimilars that registered in the Egyptian market with cost saving between 30 to 40% with an average of 35%. We should take in consideration that biosimilars are a new class of product that may be concerned that the stockholders are not familiar or comfortable enough to use biosimilars. Physicians and payers play important role in biosimilar acceptance and usage, and the patient need assurance that biosimilars are as safe and effective as reference biologics. Finally, biosimilar will create long-term cost saving and efficiencies, free up resources for other important aspects of cancer disease, cancer care, and other non-communicable disease. In doing so, biosimilar could expand cost-effective care to millions more patients, a much-needed step in reducing spending. Thank you.
Thank you, Dr. Mahmoud, and it's a pleasure to me to introduce the introduction. The second session is the introduction to biopharmaceutical medicine, which is by Professor Wolf.
How to switch on? Oh, there it is. Just here. Okay. I don't think. Yes, good morning, ladies and gentlemen, chairpersons. I have been asked to give a short introduction to those of you who are not so familiar with biopharmaceutical medicines. This may be boring for those of you who are professionals in this field, but I thought it might have a value to give a short review, a tour d'horizon, which is of course not exhaustive, about what biopharmaceutical medicines mean. Well, I start with a quite unpleasant statement. This is that most of the approximately 30,000 known diseases are still uncurable. Among those are heart disease, Alzheimer's disease, diabetes, Parkinson's disease, and so on. The therapy mainly directs into treating the symptoms and requires a lifelong therapy, or medication. But in only few cases you get a really true cure. Mostly the curing is left to the body itself. Well, why is that so? And this is so because the complexity of the human body and its cells. The human body consists approximately of 30 trillion cells. This is a 3 with 13 zeros. This is an unimaginably high number of cells. And each cell contains a complete master plan for the human body, the genome, and it represents the subplans for the individual tasks, the genes coded by DNA. And during development, it grows from the fertilized egg to the adult. Only specific parts of the master plans are read and realized in the different parts of the bodies, leading to many different tissues like tissues and cells like skin, brain, bones, and muscle. Well, the master plan, as I mentioned, the genome consists of more than 21,000 subplans, genes. And it was in the beginning of the 90s when people thought this number would be much higher, in the range of 100,000. But with the Human Genome Sequencing Project, it came down to approximately 21,000, but many more genes which are not active and which functions are not clear. And the coded information of these genes is translated into more than 21,000 proteins, which in part are then further processed to yield much higher number of different but very specific protein molecules. And the entirety of the protein is also known as the proteome. And on the right side, you see a large ribosomal subunit. This is a very complicated structure, as you can see, and it is a part of the protein synthesis apparatus of each cell. And imagine this complicated apparatus you find of approximately 100,000 to 10 millions per human cell, per mammalian cell. So this gives an impression about the complexity of the subject we have to deal with. Well, failures in the plan or the implementation and translation of the plan may happen. It could be by inheritance from parents to the child, or by external influences like radiation or chemical substances. And this could result in the omission of a certain factor. You're familiar with diabetes, when the insulin is not produced any longer, so it's the omission. It could be a negative alteration of a function, like a chronic inflammation, like in polyarthritis, you know, the inflammation of the joints is a chronic one. And, or it could be an adverse property which is acquired, like the loss of controlled growth in tumors, which finally leads to the disease. So therapy of such errors, the true therapy and cure, may require the replacement of such missing factors, as we have it with insulin, the modulation of a function, like we can do it with certain antibodies, like Humira, for example, which was mentioned before, or it could be stimulated by, if you like, to have an immune reaction against a tumor. And also, most recently, a repair of the defect can be done via gene therapy. We'll hear more about this later on today. And biopharmaceutical medicines are developed to meet all these requirements. Well, we had already a short definition about pharmaceutical biotechnology. Mine is even shorter. Biotechnology is the use of microorganisms, cells or parts of cells, for manufacturing of products. And for pharmaceutical biotechnology, you do this for therapy and diagnostics. And recombinant DNA technologies are possible because, and this is an important factor, the genetic code of the master plan of organisms, the transcription, the readout of subplans into a messenger RNA, or the translation of the subplans into functional proteins is valid and understood by all life forms. It's the same for man, for animals, for plants, for bacteria, and even for for viruses who use the same mechanism. The DNA, if you have the master plan of an organism and you want to transform it or have it producing another protein, then you can transfer DNA, the subplan coding for the very protein, you can transfer this into the target cell with different methodologies. Here you can see you have the plasmid DNA. There's no pointer here, sorry. The plasmid DNA, which requires a vehicle to be transported into the cell, and then finally in the cell, this DNA, this instruction for the protein is it either migrates into the nucleus and is incorporated into the nucleic, into the general plan, the master plan, or it stays for a while in the cytoplasm, is read up, translated, and later on metabolized. Well, most of the biopharmaceutical products which are on the market today are proteins, and they are being produced in vitro in the lab. The DNA is transferred into a target, into a manufacturing cell. This could be a microbial or a mammalian origin for industrial manufacturing. So you inject it or you transfer it in another way into the later production cell line, and then you can expand these cells. They can be kept in a nutrient medium where they start to divide. You can freeze them for storage, or after that you can thaw it. You see here the cryoculture, which then is thawed. The cells start again to grow, to divide, are transferred into a small culture vessel, and because they are growing and becoming more and more, they are then later on transferred to bigger culture vessels, which are called bioreactors. And the cells produce then the protein of choice into the nutrient medium from which it is purified later on. You see at the bottom of the slice, and from the nutrient medium to get a highly purified pharmaceutical active ingredient. And in the meantime, there are other ways to benefit from the transfer of DNA. So you could transfer DNA ex vivo when you get cells, blood cells, for example, from a patient, or bone stem cells from the bone marrow, and you transfer DNA of the therapeutic protein, coding for the therapeutic protein, into these cells in vitro, and then you re-infant these cells into the body where they do the job and hopefully cure the disease. Or, most recently, you could do this in vivo, where you package the DNA into shuttle vectors, which are virus-like structures, and then inject it into the patient, where it moves to the target tissue. The virus-like particles then somehow fuses with the target cells, and the DNA starts to be transcribed and translated later on in the target organism, in target tissue. What now makes a difference between chemical drugs and biopharmaceuticals? We heard a little bit about it already. The conventional chemical is synthesized in a chemical production plant, and you get the conventional pharmaceuticals. And the difference here for the biopharmaceuticals is that you normally use genetic engineering. These products are synthesized by the recombinant cell, and you get the biopharmaceuticals from the cell. And the difference resides in the complexity of the manufacturing process and the active pharmaceutical molecule. This gives you an impression about different molecule sizes. It starts at the left bottom with aspirin, the active ingredient of aspirin, which consists of 21 atoms, which can be easily chemically synthesized. The same is true for the blood pressure agent, an ACE inhibitor of 62 atoms, already with cyclosporin, which is used to avoid rejection of transplants in patients. Already you need a native fungus for the 196 atoms. For insulin as well, you need already an organism to synthesize it with its close to 800 atoms, and an antibody is so complicated in its structure that it never could be synthesized by chemical means. You need an organism. Mostly you use mammalian cells for this, recombinant mammalian cells. You can also demonstrate the complexity. The aspirin you can compare with a scooter, the ACE inhibitor like a bicycle, the insulin is more like a car, and finally the big aircraft resembles to the antibody, but with the difference that the aircraft can be assembled by man and the antibody needs the human or the mammalian cell. Well, also molecular biology provides new tools for targeted therapies because of the identifying and understanding of the underlying specific molecular processes in the disease, like the changes in gene expression, which I meant, opens a chance for precise, tailored therapeutic approaches. And addressing those specific alterations of the individual disease in the individual patient results in a much higher success rate and often also in a much better tolerance. And finally, in cancer, targeted therapy works by targeting the cancer-specific genes, proteins, or the tissue environment that contributes to cancer growth, like blood vessels, which foster the cancer and help the cancer to survive. This is already what we heard about. What is the difference between generics of the small molecules in the conventional pharmaceutical industry? There you have really identical molecules. It's like a mirror of the cat, of the copycat, so to say. And if you deal with biosimilars, because of the high complexity of the structure, proteins, you never can say with absolute certainty that two molecules produced with different processes are completely identical, and therefore you speak about biosimilar and not about biogenerics. And we heard already some figures about the market. The market today, the biotech market today, consists mainly of protein products like insulins, interferons, antibodies, but with an increasing number of vaccines, of cell therapy products, and gene therapy products. It grows double that fast as the conventional industry. It contributes to more than 20% of global pharmaceutical revenues. We heard about already the global sales, which are more than $200 billion per year, and is forecasted with something between 400 and 500 billion in 25. And 50% of the pharma R&D projects are biologics. But, of course, they are highly priced. You mentioned this already in the previous talk. The daily medication costs in the US have been calculated to be more than $45, while less than $2 are necessary for conventional therapies. But I think the cost of medicine is only one side of the coin. Of course, we attempt to reduce the production cost. Biosimilars is one approach to reduce costs. Shortening development times and efforts would also help, but also other factors contribute to the benefit of a therapy, like less treatment failures, for example, because you have a more targeted therapy. You could minimize the time of hospitalization if you have a better therapy, a better working therapy. The work time lost, the work time of the patient lost is being reduced. The quality of work may be improved. Also the work productivity. Job losses may be avoided, and a reduction of nursing efforts up to improving the functional status, to reacquiring autonomy is possible and should also be put on the balance if you talk about the costs of a therapy. Well, finally, with the help of pharmaceutical biotechnology, it is possible today already to manufacture industrially high-potency medicines, which could not be produced by conventional methods, to treat, cure, or prevent more diseases than ever before with less side effects, and to target specific diseases and patient groups through the use of innovative diagnostic technologies, and finally to act early on emerging diseases instead of just reacting of symptoms. And I want to conclude with a statement that with the help of pharmaceutical biotechnology, many more tools are expected in the future for better prophylaxis, precise diagnostics, and effective, well-tolerated therapies to the benefit of all patients, young and old. Thank you.
Thank you very much, Dr. Wolfer, for this valuable presentation. And now I am pleasure to introduce Dr. Bardisi to the session which address 15 years Egyptian industry experience case study. Just here. Please, yeah.
Laser pointer. Good afternoon, ladies and gentlemen. Mr. Chairman, Dr. Mahmoud Yassin, Madam Chair, Dr. Rasha Ziada. My presentation today will be addressing some misconception in the Egyptian market, which is related to the status quo of the biotech industry in Egypt. Some misconception also related to differentiating between the manufacturing of or fill and finish of biologics as compared to manufacturing from the cell to the protein. So the presentation will try or attempt to answer the related questions. It will be addressing the theme from a multidisciplinary perspective, from a 20 years plus of experience in the pharmaceutical and biopharmaceutical industry, experience in complex decision sciences and artificial intelligence. So it's a slightly different perspective than the two valuable presentations we had before. The presentation overview will start with the necessary pharmaceutical foundation of establishing a biotech operation in emerging markets as a prerequisite to merge from pharma to biopharma. A merger from pharma to biopharma will pass across certain critical biopharma milestones to ultimately be consolidated in an integrated biotech business model. Of course, it's expected to meet the related challenges, and that overcoming such challenges, or at least in part of them, will end up with the excellence model of innovation and affordability that I wish audience to be totally aware that does exist in Egypt since 14 years, or 15 years or more even. So the whole pharmaceutical foundation as a start of a biotech model started in the 1950s. That was via Heliopolis Pharmaceutical Laboratories. Those were nationalized in the 1960 and the restart of operation in Mina Farm in 1992, differentiating into a specialty pharmaceutical company with dedicated lines in gastroenterology, cardiology, orthopedics, and urogynecology. The start of the company, its pharmaceutical foundation, started via partnerships and license agreement with big pharmas and medium-sized pharmas, which definitely enhanced the quality system and the operational excellence necessary for the pharmaceutical foundation. As I said, a prerequisite for merging from pharma to biopharma. So now we move to moving from pharma to biopharma, and here Mina Farm utilized its international operation platform, starting with a joint venture with Rhine Biotech that was as early as 1998. This was the first joint venture that was aiming to implement the first biotech manufacturing plant in Egypt, Africa, and the Middle East. We're talking here about 1998, where the first joint venture and tech transfer agreement has taken place, as early as 1998. In 2001, the factory was constructed, ready to operate, and a subsidiary, which is the joint venture between Rhine Biotech and the German company in Düsseldorf, now Dynavax in the US, joined together with Mina Farm to form Rhine Mina Farm, and as you can see, operating the first biotech operations with the different setups and necessary setups to start such a complicated operation. In 2005, we have established the first R&D community, which was, of course, composed of German biotech experts and young Egyptian talents. From 2005 and 2000 to 2006, we started the launch of the first, at that time, revolutionary interferon, or pegylated interferon. That was the third pegylated interferon worldwide. Then Thrombex, which is an antithrombotic used to prevent thrombotic complication in hip and knee replacement surgery, and its topical formulation. Then came an important milestone in the history of Mina Farm, and that was the acquisition of ProBiogen, a Berlin-based leader in cell line engineering up to GMP manufacturing. Prior to that, we were a client to ProBiogen, and they did for us the technology development and tech transfer of Gonapure, which is the follicle-stimulating hormone. That was the last product that was launched in the Egyptian market for the treatment of infertility. Now, it's probably important for anyone to know that embracing a certain risk effectively in technology-intensive organization is very essential, and that the first step is to acknowledge the possibility of project failure and develop an attitude within the organization that project, unlike small molecules, are likely to fail. So developing such attitude is of crucial importance. Well, then comes some complex probabilistic models that can assess the risks, but the complexity of the biotech industry and its evolution across the last 2 decades has made such models, mathematical models, not really effective, and it became up to the executive to do just 2 types of differentiations. One is the level of the risk, starting from low, medium to high, and the second thing would be looking at the risks from preventable risks, that means within the operation itself, or strategy risks, which are the risks that occur due to taking decisions and making high investments, depending at the time of the investment for the sake of reaching the goals and reaching high revenues. The third are the external risks, and these are beyond the control of the company. So if we look at here, this is the NASDAQ composite and a reflection of the performance of the biotech industry starting from the 1995 through to 2017. We can see that the tech transfer agreement that we did with Rhine Biotech was at a time of poor performance of the NASDAQ, and hence reflecting poor performance of the biotech industry at large. And that was definitely a period of high risk. So we took that decision during a period of high risk. Then there came the bubble, the famous biotech bubble that took place between 1997 and approximately 2001. This period, and afterwards still there was no outstanding performance of the biotech industry, but then comes the acquisition we did at ProBiogen, also at a time of bad performance of the biotech industry, after which there was a surge in performance. So most of the decisions we took here were at periods of high risk. So acquisition of ProBiogen is a renowned Berlin-based technology provider and developer of biopharmaceuticals, a leader in cell line development, cellular engineering up to GMP manufacturing, and provider of proprietary technologies. Now I'll just give you a short or a synopsis on the technologies that ProBiogen has, and that are becoming very important technologies in the biotech industry right now, and are being licensed to world leaders in biotechnology in the industry. Glymex is a glycoengineering technology in which the natural killer cells are enhanced, and this is used through afucosylation. That means removing the fucose from the N-glycans of the proteins in order to enhance the natural killer cells' ability to be attracted by the immunoglobulins or antibodies to attack the infections, the tumor cells or infections, and this technology allows 100% more activity, up to 100% more activity in dealing with tumor cells and infections. Then there's the revolutionary transposase technology. This technology is becoming increasingly important, since what it does is that the cell line development, one of the specialties of ProBiogen is usually used on or usually does the transgene integration in a random fashion, but the transposase novel technology did in a way radically change this approach to just integrating transgenes in the areas or in places where there is the highest transcriptional activity of the genes, so providing higher yields and driving the costs. Then there's the AGE technology, which is a new expression system for expression of vaccines, like the influenza vaccine, like the vascular stomatitis vaccine, and like the herpes vaccine. Now, combining the transposase, the AGE technology, and the expertise in viral vector technologies has also put the company at the forefront of being a possible provider of solutions for one of the industry's main problem in gene therapies now, which is the yield of production. And last is the human artificial lymph nodes, which are 3D microorganoid models that simulate the body's immune response, or not the body's, the lymph nodes' immune response. They're now part of a huge program to put all the organs of the body on a chip. So like exactly an electronic chip of a computer, have the organs, the lymph nodes, the heart, the blood, to simulate the whole body for early drug development. Now, ProBiogen has a very interesting selection of global customers. You can see the world leaders of the industry are using the services and technologies of ProBiogen. And for example, the Glymax technology I was just talking about was licensed to companies like Roche, like Bayer, and like Sanofi and other companies. So, of course, once an Egyptian company acquires a modern biotech leader in Europe, that is very unusual for the Europeans. So the milestone itself was definitely very much triggering the interest of the press in Europe to see that happening, an Egyptian company from the emerging world acquiring a biotech company that is offering services to the world leaders in the biotech industry. You can see the reaction of the press there. Now, the result of all those milestones was an integrated business model. When we started with a joint venture with Ryan Biotech, we had a technology transfer. Then we developed the product further clinically and reached to marketing 4 of the products. When the acquisition of ProBiogen took place, there was a backward integration starting from cell line development and all the necessary related technologies. So all of a sudden in Egypt, we had a business model that is unprecedented, starting from cell line development through to marketing of the products. Now, as a result of that, the different phases, starting from the tech transfer to acquisition of ProBiogen, we had 3 products launched in the market already. We have other products in the pipeline, among which 5 are monoclonal antibodies, and they are expected to be launched from 22 to 28, mostly addressing autoimmune disease and cancer. And here we have to, of course, think that this is not a fill and finish process. This is a process of manufacturing from the cell to the protein and might result in products that are really reaching a reduction of the price of the originator up to 6 folds. And this is what we encountered with previous products that were launched. So the outcome was the launch of those proteins, the development of a rapidly growing pipeline portfolio, as you can see, and offering the market cost-effective products that are, again, developed from the cell to the protein, not just imported products that are filled and finished. We tried as much as possible to ensure the safety, the reliability of our products, in addition to cost effectiveness, and had a lot of literature and publications in international journals about the efficacy and safety of the product. And as you can see here, that was one of the WHO's publication showing that Egypt's cost was reduced via our pegylated interferon to be the most cost-effective product and compared to other countries in the world. Of course, you're expecting to meet challenges when you are embarking on an operation like that. So we had industrial culture expertise and talent challenges, development costs, regulatory challenges, marketing and distribution challenges. I will address them in short. One of the main, they are in descending order in terms of becoming less important as we go down. The most important was the absence of a biopharma culture. We are in an emerging country, and of course the talents and the pool of talents required to have such a project is not an easy task. So we could overcome that via the tech transfer partnership, the presence of expats, the merger between the pharma and biotech. Of course, while it is acknowledged that there are differences between both industries, but they definitely share commonalities. And what we did was an attempt to merge the pharma and biopharma personnel and get them acquainted with the new industry. And cultural hybridization occurred through making synergy projects such as with ProBiogen, with experts coming from Germany to Egypt and vice versa. Here you can see the Egyptian team. Here you can see the expats. Here you can see the German experts from ProBiogen. And you see at the later development phase, a lot of German experts who are coming and Egyptians going to Germany. And this process continues in order to overcome the problem of creating that biotech culture among the scientists in Egypt. The development costs, as you can see, of course, there was a surge following devaluation, 2 to 3 times the cost of any project prior to devaluation. There's always an inter-project variation, so you cannot really project well from a eukaryotic cell expression line system or a mammalian cell system. A difference between the different molecules have different development costs. There are projectable costs of preclinical till phase 1 that are supposed to take place in Europe according to Egyptian guidelines, so those are projectable. Of course, still there remains some uncertainty regarding the projections of the cost of phase 3, since we are boarding on that at that point in time for the new generation of monoclonal antibodies. So according to regulatory channels, initially, which is normal, there were some difficulties in communication with the regulatory bodies, but progressively there was a rapid improvement and better communication. Still, there are some minor issues to be solved, such as R&D department logistics. They are still not regulated, so an R&D department that exists in Egypt still needs to be acknowledged. And the biopath for phase 3 is probably described, but there are some issues that need to be clarified, especially regarding the originator products that need to be procured in order to be able to carry out phase 3 studies. Of course, marketing is very interesting once you have developed a product and you've gone through the most complex parts of developing the product. You have such a sense of security. You believe that everything has been resolved. Now it's time to go, but that is really a full sense of security, because what happens is that there's a fierce post-marketing competition, often that going beyond some ethics of marketing. Of course, you're competing with big pharmas, and such occasional unethical behavior passes unpunished. So one should expect fierce competition and be ready to deal with that, because having a product on the market is not the end of the journey. The distribution challenges, of course, these are temperature-sensitive projects, and we did, from the beginning, start by using indicators that ensure that the product has been stored and transported under controlled temperatures, and develop that further with a computerized system that can measure humidity, temperature, and see where all the transportation, all the cars are, and record that online on real time. So ultimately, what resulted was an excellent model for innovation and affordability. With its core, we have the process R&D. This is the ultimate and how it ended the model that we have, an excellence model of innovation. So around the R&D, process R&D, making products affordable and innovative, we find the manufacturing process operations and according to GMPs, GLP and GDP, interacting in a constant interacting mode with the R&D department, all embedded in a quality system that definitely benefit from the multi-alliances and the multi-alliance pharma model they had with 40 tech transfer of small molecules, but they still helped in development a quality system that can accommodate sterile production and be ready for the fill and finish of complex molecules by recombinant molecules. Then, of course, the technology, the manufacturing operation benefited from the technology transfer from Germany, from RAN Biotech, with the launch of the 3 products. We built during that time an Egyptian-German biotech excellence network for suppliers doing consultancy, service suppliers, consultancy, doing comparability studies and formulation studies, which are rather much more difficult for the recombinant products. We could reach a sort of cultural transformation, get the necessary culture for the biotech operation, and of course got access to novel technologies that I've give you a synopsis about before, to then be able to comply with regulatory distribution logistics, as well as the clinical research. As I've seen, we've published in many international journals. We then proceeded and are very much focusing now on the data integrity that is becoming a very important issue according to European and FDA guidelines, embedded in an ERP solution and going to the market with a product. So 3 products on the market, 1 topical formulation that is the first in the world right now. And of course, you can expect such an excellence model for innovation and affordability to be acknowledged worldwide. And you can see here the Newsweek talking about MENA Farm's role in the biotech industry in Egypt, the Times, and last but not least, that was a few months ago, a supplement by Die Welt that was basically just getting examples of the mega projects taking place in Egypt in the field of energy, electricity, etc., but did not forget to include the successful model of MENA Farm and its role in the future of the healthcare in Egypt and also in the Middle East. I conclude. A robust pharmaceutical foundation is essential for embarking on biotechnology. The merger of pharma and biopharma is a very complex process that needs a cultural transformation that took us about over 15 years. There are risks and challenges that should be stringently dealt with. The business model of this case study is atypical, so it's not a typical model, but can be helpful to build other successful models. I'll finally end with a short film, about 1 minute or more, about what's happening really inside MENA Farm. So what you're going to see is reality, apart from some animation. It's happening inside MENA Farm. So can we have that film?
Lives and winning the battle of fighting diseases are the aim of medicines. Standard therapeutic approaches use simple chemical compounds. Despite clinical advances in many diseases, cure remains the exception and not the rule. Recent advances in medical research have resulted in a paradigm shift in the treatment of disease. MENA Farm was an early recognizer of this shift, initiated a transition towards complex molecules, establishing in the mid-90s the first biotech facility in Egypt, Africa, and the Middle East. Advanced biotechnology, the next generation medicines, use the body's immune system for its precision and ability to recognize and attack malfunctioning cells, founding what is known today as immunotherapy. Immunotherapy implies the use of complex medicines to program the immune system to use its amazing ability to target and kill affected cells with minimal side effects. At the forefront of advanced biotechnologies, together with its acquired German subsidiary ProBiogen, MENA Farm is manufacturing 5 innovative immunotherapies to treat various challenging diseases such as cancers and rheumatoid arthritis, making them affordable for patients in Egypt and emerging countries. MENA Farm. Innovation made affordable.
Thank you, ladies and gentlemen, for your attention. I just want you to remember that the biotech industry exists in Egypt since 14 years and will continue to thrive. Thank you very much.
Thank you very much for that the very valuable steps and history and presentation from Vina Farm, Professor Dr. Bartisi. And now we will share with Dr. Medhat Ughbashi the academic industrial collaboration supporting local biopharmaceutical industry from the academic point of view. أهلا دكتور.
Good afternoon, everybody. First of all, I would like to thank Mina Farm and Professor Mahmoud for the invitation, for this invitation. It's a pleasure to be here among my colleagues and professors and present an additional piece to this successful model, where after having the opportunity to adopt such model in Egypt with successful technology transfer, it was the perfect time actually to get the academia from the Egyptian society involved as well. My talk today is a bit different, but actually it is very complementary to what Dr. Mahmoud and Dr. Wafiq presented in the previous presentations. My talk is about academia-industry collaboration supporting the local biopharma industry. Actually, I have added this little phrase after it. Are we there yet? I'm not quite sure, but this is one of the initiatives that has been established. It's very similar to experience of Mina Farm. They are playing solo in this sector in Egypt, and I assume we are also doing the same perfect job from the point of view of the academia. Here, one of the best case scenarios where the academic research can lead to a Nobel Prize. Yes, this is Jim Allison, and his work ended up in the market with a novel treatment for skin cancer as well. So it's one of the best case scenarios. We hope to be there someday. We do share common goals between the universities and the industry. Actually, these goals go hand in hand. First, we are the responsible party for producing talented graduates. Talented graduates are those who will lead the industry and the regulatory framework in this sector. Second, we have the opportunity to invest in research and innovation. The industrial sector has the same opportunity as well. So we do meet here with the collaborative research projects such as the one I'm going to present. Finally, it's a social responsibility. Both of the universities and the industrial sector share this responsibility as well. We are all serving this community. The industry is part of the community as well, so we need to support it in all possible means. This actually met kind of good environment to foster our intent, the shared intent between us in the university and Mina Farm, where Ministry of Higher Education, represented by Science and Technology Development Fund, allocated a funding opportunity for demand-driven projects. One of the demands for this community is technology transfer. So we decided to go for some additional money to be able to invest in the R&D of this industry. The main conditions required were we have to show a solid collaboration between academia and industry. Second, the human resources that could trigger and implement this project. Third, the project proposal itself. The first item, we need to show that the academic research we are going to do will support the industry in developing their own products, and their product is very likely to go into the market. Actually, there has been a lot of research, even with industry, but just the role of industry was in-kind support. In-kind support means, okay, some of the researchers from the universities can go there, do some experiments, they can get some fund through the industry to achieve their experiments, but end of the day, nothing reaches to the market. And on the other side, the industry has to share funding. This is very, very, very difficult situation. If you are coming from academia and wants to convince an industrial partner that your research will support their plans, they will eventually be able to share the co-funding. And this is the actual scenario we implemented. For the human resources, we have talented scientists in both the academia and the industry, well-trained scientists, and also we have to show that this project will support junior scientists. Junior scientists, I mean by them, those getting their master or PhD degrees from the university, or even those working in the industry, in the R&D. They do not need to have a degree, yes, but they need to show competency in research as well. This is an industry that is based on research, and the technology changes very frequently. So they need to be very up to date with such changes. With respect to the project proposal, we need to show that we have a market analysis there. This market analysis will actually coincide with what Professor Mahmoud presented, because it's something very trivial that we have to do before we start. The second thing is to show a business plan. This business plan should be sustainable. And by sustainable means, yes, it's not just an endeavor. We are going to have this once in a lifetime, and then we could not repeat it. No, we are going to have this project, and this project will lead us to the next one, and after it, we can establish a sustainable facility. Our project is about the assessment of biosimilarity and interchangeability of locally produced biopharmaceuticals. So we had this initiative originally started at Cairo University, but we applied for this project through New Giza University and our colleagues there. We collaborated with Mina Farm because they have this high tech in their facility, and they were willing to collaborate and provide the materials to apply our experiments on. My bioanalysis research group, actually I will come back to it later in the last slide, because without them we could not have achieved anything from what I'm presenting now. For the market analysis, of course, the biologics are the future of pharma industry, and these are the data extracted in 2018 when we were assembling our project. So we have 11 out of 15 top-selling drugs are biologics. Out of the 11, 8 of them were monoclonal antibodies. So we kind of defined the target for our study. We are going to invest in monoclonal antibodies. Again, the cost versus access affects the access to medicine. So having the opportunity to produce biosimilars will serve the community, which is an integral part for any assessment of these projects. Here we have a list of products, the top-selling ones, losing exclusivity. So we need to pick one that could serve the community and meet also the production plans and growth plans at Mina Farm. We focused on breast cancer simply because it's the most commonly diagnosed cancer in female worldwide, and also in Egypt we have a report that the incidence rate reaches 32%, with an expectations to rise up to 1.5 folds in 2050. And this is also about breast cancer. it's a leading cause of death in females. So we identified the disease that if we started to produce a biosimilar to treat it, we will end up with a good service to our community. Here, we picked trastuzumab. Trastuzumab is one of the best-selling drugs, $7 billion US dollars a year, and also the exclusivity was about to expire and expired in some other places, so it's a perfect candidate. It's a monoclonal antibody, one of the best-selling in the world, and it treats one of the novel treatments for breast cancer, which is a major issue for the Egyptian society. The status at that time, and actually it has not changed a lot, there are about 18 biosimilars either approved or in the pipeline worldwide, and in Egypt we have around 3. They are at different stages of registration. This is the design for our project. Again, I recap on what Dr. Mahmoud presented. Yes, the analytical part and the analytical comparability study takes a lot of work when it comes to production of biosimilars. So this is just a little list of the equipment and techniques that we need to do to prove that any newly produced biosimilar is actually a biosimilar molecule to its originator product. It ends up with some cell-based assays and functional assays, so we need to correlate these results all together. Here, it's the core of our study. Totality of evidence means that we have to get the data from different techniques, all the previously presented ones, in addition to the cell-based assays and functional assays. We will share this practical part with our colleagues in MENA Farm. So we do have the capabilities to cover part of these experiments, and they do have the rest, and through collaboration with the German partners in ProBiogen, so we can achieve the rest of these objectives. So we have the orthogonal testing, which is kind of collection of every possible technique that can look at the molecule from different angle, and then the physicochemical characterization, and ending up with biological characterization. Having such data from all these techniques presented in the previous figure, then we can end up with the totality of evidence. Totality of evidence, that means that we cannot conclude biosimilarity using the results of only one technique. No, we have to look at the molecule from different aspects using different technologies. These technologies should not be similar to each other. So just go to the analytical chemistry bit a little bit. So if you have an HPLC, you cannot say that an assay using a reversed phase with C18 and another assay with C4, they are orthogonal. No, they are actually in the same principle. They are both based on the hydrophobic interaction. No, you have to incorporate those based on the hydrophobic interaction, charge, size, glycan analysis, every possible assay you can achieve. Additionally, we have incorporated a little bit of innovation here. We are actually working on transforming the qualitative data that can be obtained from techniques like peptide mapping into quantitative ones. This comes through integration of chemometrics. Chemometrics are kind of analytical techniques, yes, but they are more concerned with the complex mathematics, and they need specialized softwares to do. So if you end up with a peptide map and you want to compare the peptide map of the originator to that of the biosimilar, you can do it visually based on the retention times. That's all what you can do. And then it's kind of fingerprint for you. But if you can transform this data mathematically into a meaning score, and then the score can be surrounded by plus-minus range, so you can compare these data or these maps statistically. And this is what we actually started to invest in through doing peptide mapping and chemometrics for a small It's not small molecule, but it's a growth hormone. Growth hormone, it's a protein, small protein. It's not really comparable to something very complex like a monoclonal antibody. But we were able to show that we can compare the two molecules to each other. We can detect differences from batch to batch. We can detect the differences related to the formulation additives as well, and we can identify the source of differences. We can also detect any degradation that could happen to this molecule. So having this model, we were able to develop purity and stability indicating assays. It's kind of another expression for the totality of evidence. Instead of having a huge set of techniques, we can have one technique, even HPLC with a UV detector. It's not a fancy equipment like a mass detector or something like this, but having the data transformed mathematically can give very useful information. We can even detect changes in one peptide in the peptide map. If we have just one amino acid difference due to mutation in the host species producing the molecule, this will affect the peptide fragment on the HPLC chromatogram as well. We did this already for the peptide mapping, and we are extending this application to glycan mapping as well. Eventually, in order to satisfy the need for sustainability, we are going to have these technologies and these techniques at university in the form of a research center that is specialized in such tools. So, in order to answer the very first question, are we there yet? Actually, we need to do a lot of homework. The first thing is, if you want to work with industry, you have to be fully aware about the registration requirements and the regulatory framework as well. You can do fancy experiments in the lab. End of the day, these are not the ones required for the manufacturer to proceed. They could be a very good publication, but you might not be able to support the production in the manufacturing site. Alternatively, if we adopt the regulatory framework and its requirement right from the beginning, we will end up with a good publication again, but with results that could be submitted to regulatory authorities. And one more thing, I will reflect on what Dr. Rafiq presented about data integrity. Every equipment we...
activities and the goals you have for Egypt. So let's switch gears a little bit. We heard very much about biotechnology, biosimilars, and all the science, and now we are entering, and it has been mentioned by my previous speakers, into the world of regulation, which I guess for some of you is a kind of black box maybe, I don't know, and let's see whether we can lift it up a little bit. So the Paul-Ehrlich Institute is a federal institute for biomedicines under the Federal Ministry of Health. This is our current Minister of Health, Jens Spahn. And the Paul-Ehrlich Institute has been founded in 1972 as the Federal Agency for Sera and Vaccines. And since 2009, we are the Federal Institute for Vaccines and Biomedicines. Within Germany and the European framework, the Paul-Ehrlich Institute here, you don't see the pointer on the left-hand side, is one institute of others under the Federal Ministry of Health regulating medicines. We have a big sister agency which is in charge of all the pharmaceuticals, which is called the BfArM, and they also have our information system there, which is called DIMDI for medicinal products, and we have a Federal Center for Health Education under the Ministry of Health, and we also have the Robert Koch Institute, which is our public health institute of Germany. And of course, since we are a regulatory agency, we work closely and we are part of the network under the European Medicine Agency. And there we have representatives in the CHMP, as we call, or the VMP, which is the Committee for Medicinal Products for Human and for Veterinary Use. So here you see the pictures of our current president, Professor Klaus Zichotek, and our vice president, Professor Stefan Fietz. The structure of our agency is summarized here. So we have the management with our president you just saw. We have our own research groups and coordinations, which are linked to the presidents and the vice president. We have units of the management, what we need, and then below you see the different departments we have with administration, major policy issues, and coordination. This is the department where I'm located and my project is located in. We have departments for safety of medicinal products, and then the more product-related departments for microbiology, virology, immunology, veterinary medicines. That's what we are also responsible for, allergy and biotechnology, as well as blood, hematology, and transfusion medicine. And below you see general groups like the staff representation and temporary research groups and professorships. So what are we in charge of in terms of regulation? So we regulate vaccines, that's a part of our name. SERA, like immunoglobulins and monoclonal antibodies, including biosimilars. Test and therapy allergens, blood and blood products, tissue preparations, and then so-called advanced therapeutic medicinal products. And I mentioned that already, vaccines and SERA also for veterinary use. What is our responsibility with regard to the development of marketing authorizations and the development of new medicinal products? So it starts with a discovery at the very far left, and then going through the manufacturing, non-clinical study, clinical studies, and then in the end, when it is on the market, then also the kind of life cycle management through variations, batch release, and so on. And through the discovery, from the discovery up to marketing authorization, we also have tools in place or interactions with the discoverer and the manufacturers in terms of scientific advice, and also we are in charge of approving clinical trials. We also have, and this is something going back really to our founder, Paul Ehrlich, that we also conduct research at the Paul Ehrlich Institute, because we think that especially biological medicinal products are complex, and we heard that. This is also a very important topic in developing biosimilars, the complexity of biological medicinal products. And it was already the idea of Paul Ehrlich that you need to do your own research in order to understand these products and be able to properly regulate them. So the three main areas where we conduct research at the Paul Ehrlich Institute is, of course, regulatory research with regard to innovative medicines and product testing. We look into pathogen, host, and biomedicine organism interactions, and we also have research with regard to experimental vaccines, therapies, and diagnostics. We do not do product development. This is not part of our mandate, since we are a regulatory authority. This is basic research which gives us the knowledge and the possibility to learn from research how we best can regulate medicinal products. As a regulatory agency, we are also part of an international community, and we are working very closely with the World Health Organization together, and we have two WHO collaborating centers at the Paul Ehrlich Institute. One is, that's the older one since 2005, we are WHO collaborating center for quality assurance of blood products and in vitro diagnostics and in vitro devices. And we have also a WHO collaborating center for the standardization and evaluation of vaccines. And since a couple of years, since almost 5 years, we also have a program under the Ministry of Health, based on the decision of the German Parliament, which is called the Global Health Protection Program, where I also lead one project within this program, and I will talk a little bit about that in the second part of my presentation. So with regard to the WHO collaborating centers for blood products, there we support WHO in developing international standards. We conduct laboratory studies in order to test these international standards, and we support pre-qualification, and we were also quite supportive with the World Health Resolution 6312, where it was about to list blood products also as essential medicines from the WHO. The way we work, for example, here in the WHO Collaborating Center for Vaccines, is that we mainly work through the ECBS, which is in yellow in the center of this slide, the Expert Committee of Biological Standardization. This is the oldest expert committee of the WHO. And we are part there of a network of other regulatory agencies like the US FDA, Health Canada, NIBSC from England, TGA from Australia, MFDS Korea, Japan, China, and then you see also the Paul-Ehrlich Institute. And with all these colleagues, we support the work of the ECBS in developing international standards. And the way it is organized within the Paul-Ehrlich Institute is that our section or our department, which is called G3, this coordination of international activities, is coordinating the experts we have in the various departments of our products, which are then listed here around the G3 dot. We have other activities where we support the WHO as well. This is, for example, through the Expert Committee on Product Development for Vaccines Advisory Committee and the SAGE, where we support expert groups in order to help making international recommendations for vaccines and vaccine use. Coming now to the second part of my presentation, which is part of the Global Health Protection Program, a newly set up program of our Ministry of Health under the experience we had with the recent Ebola crisis in West Africa. And here you see my team, where we work on the regulation of biologicals with a focus currently on vaccines. The start of this program was at the G7 meeting in 2015 in Germany, where the leaders, which you see here, clearly stated that with the experience we made and the problems we were facing at the Ebola crisis, that the international community really needs to be better prepared. And what can we do? And our ministry then asked the different institutes, which are mentioned at the beginning, like the Robert Koch Institute, us and the BfArM, what can we do in order to help being better prepared for future international health crisis situations? And we were making different proposals, and this program then developed further. It became bigger and bigger, and since 2019, this program is now a program of 24 different projects and is working with 34 different partner countries in Africa, Asia, and Southeast Europe. And on the right-hand side, you see the German institutes which are conducting these projects. It is our sister agency for pharmaceutical medicines, the BfArM. It is our Bernhard Nocht Institute, it's the Tropical Institute for Germany. It's the Robert Koch Institute, our Public Health Institute, a research center, Borstel, focusing very much on tuberculosis, and also our Veterinary Institute, the Friedrich Loeffler Institute, and ourselves, of course, who are conducting currently these 24 projects globally. In Germany, we have two, we call them modules, or you can call them projects. One we call Blood Train for training, and the focus here is on the regulation of blood products, their safety and availability. And the other model, which we call VacTrain, because we started with vaccines, but we also expand now to biologicals, is about regulatory training and advice in the area of vaccines and biomedical therapeutic products. And we also focused at the beginning with clinical trials, but we are also expanding that now to pharmacovigilance and regulation of biologicals with regard to quality. We had a lot of interaction with WHO and other international partners, and we started our pilot project in West Africa, partially because there was this big Ebola crisis and the need in order to get support was very high, but also because of the general international agenda from the WHO, where they were conducting a global benchmarking in this area, which is very important for us and a very good tool in order to see what is the current status in this region with regard to regulatory capacity, and then how can we support that. So that was the reason why we started in this area. We were part of the WHO benchmarking of all the countries in the ECOWAS region in West Africa. We supported WHO for that. We got a baseline of what is there in terms of regulatory capacity, and we asked for applicants who would like to join us in this pilot phase, and we got very good applications then from the Gambia, Sierra Leone, Liberia, and Ghana in order to be the pilot countries for our training activities. And the countries were chosen by purpose this way, and you see two different squares here. We have the Gambia, Sierra Leone, and Liberia, who are less resourced in their regulatory capacities, and we have Ghana, who we work more closely in terms of partnership together, helping us to conduct the training in the region, since this is an agency and this is a country in this region which has more resources than the other partners, for example. So that was chosen by purpose, and you will see that in a minute. So the way we work is that, especially for the less resourced countries and agencies, we are focusing on so-called structures, because that was what we saw under the benchmarking, or as a result of the benchmarking, that there were a huge lack in regulatory structures, meaning appropriate legal framework, which is the absolute basis, then operational guidelines, which is showing also applicants what is expected in terms of documents and in terms of regulatory processes for applicants in clinical trials here. And then develop also internal SOPs and templates so that the regulators in the agency know what they actually should do and what they should focus on. And when these structures are set, and this was the focus for our pilot countries who are less resourced, you can go and make the next step and develop assessment capacity, the scientific capacity you need in order to conduct preclinical, quality, and clinical assessment prior to the approval of a clinical trial or prior to the approval of a marketing authorization. And here's an example of what we were working on, also together with WHO and a network in Africa, the AVREF, the African Vaccine Regulatory Forum, which is focusing on capacity building in Africa for the regulation and oversight of clinical trials. And we worked with them through all kind of activities, starting from the legal framework, as I was saying, the operational guidelines and development of SOPs, what is needed, with a focus, or not with a focus, keeping in mind the international standard for benchmarking of regulatory agencies, which has been developed by WHO. So we didn't develop anything from scratch on our own. We do work in the framework of international standards developed by WHO and help then the agencies to develop their appropriate structures. In general, the strategy we have with the whole project is that we build up on 3 different areas. So I was talking about building up regulatory capacities and structures, which you see on the lower left side of the slide, which we did with individual countries, tailor-made according to their need. We are also trying to foster regional centers of regulatory excellence. That's why I was mentioning Ghana as a more partner for our work than the work we do with the less resourced countries, so that we support regulatory agencies in the region which are already more developed and can provide training by themselves. So that the training is not just coming from us, from the north, so that we also have the possibility and strengthen the capacity for the region itself to have a sustainable structure for training themselves and keeping their knowledge. And then, of course, what is another important pillar for us is working through already existing networks and international partners like the WHO and developing agencies in Africa like NEPAD, for example. After the first phase, that was considered to be quite successful what we did and well received, and we were also able then in a period where the program was expanded by our ministry to start a project we do jointly now. That was the first joint GHPP project between two German institutes, which is a good step, was a very good step. So between our partner institute BFAM and the Paul-Ehrlich Institute, where we used the approach we already developed in terms of supporting regulatory agencies and bring in the capacity of the BFAM in dealing with the regulation of small molecules and other pharmaceuticals in order to form then the joint project which we call RegTrain, which has the aim to cover more or less the full scale of regulatory capacity as much as we can with the resources we have, of course. With this extension, we also expanded then our reach out within Africa. Since the BFAM had already collaborations with Zimbabwe in other projects, we were also supporting WHO again with the same approach to look at the benchmarking of all the regulatory agencies in the southern part of Africa. And we choose Zimbabwe as one of those with more resources in the region as our partner, like we did already with Ghana, in order to develop a train-the-trainer program, which is bringing the agencies into the position that they build up their own trainers in order to train people in their own agencies, but also train people from other agencies, and doing that with our support. And the way we do it, and here you see a kind of summary of our project and the achievement at all, is that two people who were chosen after an application are joining us for training, and we started with pharmacovigilance this year. You see at the upper part a picture where the two trainees are sitting together with my colleague who's also here from our team and discussing things about pharmacovigilance. Of course, we are very happy that we are supported within our institute from all our colleagues and our president with our work. And we make sure, if you follow then the curve, you see that the regulators we trained, we gave support, are then also providing this knowledge to their colleagues. And I just got a very nice email from a colleague in Zimbabwe that they are very happy with the support, and we're already implementing things the trainees were able to learn during this course, and bringing the regulatory agency a huge step forward. Then you see also, if we follow this circle, all our partners, we are working closely with the African Union, the World Health Organization, NEPAD, the World Bank, and the Bill and Melinda Gates Foundation, and make sure that whatever we do is internationally harmonized and not just our individual interpretation of how we think regulation should be done. And then, last but not least, you see a very nice picture which is trying to summarize the development of structures we were able to give in our partners, the Gambia, Liberia, and Sierra Leone, based on the indicators which have developed by the WHO. So the indicators, they are, for those who are not familiar with the benchmarking system, they're addressing certain regulatory functions which need to be there in order to be a functional and competent regulatory authority. This is very, very much defined and specified. And at the first row, you see there is just one bar on the far left, which was setting kind of the basis for regulation at all, and having a very, let's say, small legal basis for regulation, and then with our individual work, we were able to support them and develop documents and criteria also to fulfill other parts of the benchmarking and the indicators. And with this, I would like to thank you for your attention. And I would also like to thank our partners from the Gambia, Liberia, Sierra Leone, Ghana, Zimbabwe for this wonderful collaboration we have, and this will continue, of course. Our partners internationally, NEPAD, AMRH, WHO, AVRF, this network for regulation of clinical trials, partners at the US FDA, with whom we work very close also because of our WHO collaborating centers. And then you see our team, colleagues from the Paul-Ehrlich Institute and from the BfArM. Thank you very much for your attention.
Okay, let's start with the presentation from Professor Dr. Duncan Craig. Yes, Lisa?
Thank you very much. Is it better to use this or these ones? Okay. Oh, right, I need the laser pointer, definitely. Right, and that's the clicker there. Okay. I'll use these, that's all right. Okay. Didn't have enough hands. Can everybody hear me okay? Yeah, okay. Well, first of all, can I say what a great pleasure it is to be here in Egypt. I come over here quite a lot and it feels like a second home. And one of my great pleasures is to put on Twitter that I'm in beautiful sunny Egypt when it's extremely cold and miserable back in the UK, to annoy as many of my colleagues as possible, and it works every time. I'd like to thank the Ministry as well, the Ministry of Health, for having this meeting held under their auspices, and also MINAFARM for inviting me to speak here, and my other colleagues and partners from other countries. Can I begin by saying that I think that this conference is an extremely good idea to have, particularly because this is clearly a point of pivotal development within Egypt. And we have an expression in the UK, which I'm sure you have something very similar in Egypt. It takes a village to raise a child. And I think what we can say is that it takes a village to raise a biotech centre as well. And perhaps the people in this room and attending this conference are going to form the future village, because we need a multitude of skills and inputs in order to make this work. So what I'm really going to be talking about in this presentation is the kind of village that the UCL has experienced. Not so much just to parade all the things that we've done in this sector, but just to show you the mechanisms and the support that we rely upon to make our own biotech activities successful. So I apologize in advance. This is slightly going over familiar territory from the presentations of my colleagues. But I think one of the key things that pretty much everybody has emphasized, and I think it's worth emphasizing again, is, yeah, that's working, is that basically what we mean by medicine has changed. As you're very familiar, in the old days, then basically we were entirely reliant on plants. We had synthetic low molecular weights. We moved into the biotech area. But really, we're looking at a whole new generation of ways in which we can treat disease at the moment. Things like gene therapy, cellular therapies, and regenerative medicine. So these are very exciting times, but also, as my colleagues have already said, this brings with it challenges that we need to face up to, and we need to realize the rules of the game are changing as well. But I think, congratulations to Egypt, congratulations to MINAFARM for showing the willingness to actually look and embrace these new opportunities for not just treating, but as colleagues have said, curing conditions. And I think for many people, this is what is really, really exciting. And I'll give you some examples of where conditions where we thought the most we could ever do is to treat the symptoms. We can now possibly look at one-dose applications to actually cure the disease for good and make it eradicated from the patient. This is not the most sophisticated slide that you're going to see in the whole conference, for which I apologize in advance. But basically, this is my sort of attempt to slightly crudely summarize the different types of approaches, and I'm sure colleagues can add on to this or criticize it or whatever. I don't really want to talk about natural products too much because I think it's reasonable to say they almost live in a parallel universe when it comes to regulatory affairs. I'm nervous of saying this with an international expert sitting to my right, but I think that obviously you've got things like traditional Chinese medicines, and so there is definitely a place for these medicines. But this meeting really is looking more at the centre and the right of the way in which we develop medicines. So clearly, a staple is the generics industry, which is in very healthy state in Egypt. We have a good relationship with Evapharm, for example. But an interesting area is what is being nicknamed super generics, whereby instead of just using conventional dosage forms, we're actually now using much more sophisticated systems, such as aura-dispersible systems or whatever, and using those to actually put a new spin on the idea of using existing drugs. Biosimilars have already been mentioned in quite considerable detail, and obviously these are going to be hugely important moving forward. And then we have the, if you like, the semi-traditional low molecular weight drugs, the biologicals, nanomedicines, and what are now being termed ATMPs, or advanced therapy medicinal products. And in many respects, the interest is really very much around these, but the likelihood is we're all going to end up with a mixed economy of all of these things, and indeed these working together for the benefit of the patient. So actually, there are many, many opportunities for advancement and looking at ways of doing things in a new and innovative manner. Now, in terms of UCL's activities in this sector, I think it's probably reasonable to say that the ones that we've highlighted in yellow here are the ones in which we've got the most focus. We do a little bit of regulatory work, but as I say, I think there are others in the room that are doing more than we are for biosimilars. But really, we've got a big interest in the super generics, but then all of these more advanced techniques. And in many respects, that is what a university is there for. We're meant to be trying to do exploratory work, hopefully at the cutting edge of all these different technologies. So what I'm going to be doing is I'll talk a little bit about my institution for those of you that are not familiar with it, and in particular how we are currently interacting within Egypt, but then I'll go on to give some examples of not only the activities and projects, but also the manner in which we are supporting these. And I think that is very, very important, because it's not just an idea, a matter of somebody very clever having a good idea. Somehow that good idea has got to get through what we call the valley of death, which is the translational process to go from the idea to something that can appear on the market. So just to talk a little bit about UCL. So basically it was founded in 1826, and it was founded on the ideas of a philosopher, Jeremy Bentham, and this is Jeremy Bentham sitting here. Now, when I say this is Jeremy Bentham sitting here, I mean that absolutely literally. This is the mummified body of Jeremy Bentham, apart from the head, which is made of plastic, because apparently the medical students used to steal it and hide it in various places around London. And he sits outside the provost's office. But he came out with an incredibly simple idea, which to us now would seem to be very, very obvious, that universities should be there for anybody that can get benefit from attending, irrespective of their gender, their race, their religion. And now we are completely used to that idea as being very largely the norm. Then in the UK, it was completely revolutionary, and it was essentially the third university to be created after Oxford and Cambridge. And really it pushed open the door for egalitarianism within the university sector in the UK. Now, I say this for two reasons. One is because I'm deeply proud of that fact. And secondly, because I think you can argue that that culture and that spirit has actually gone down through the years. And one of the things I like about working at UCL is it's a very, very collaborative institution. And yes, we have the occasional person with a big ego, but generally speaking, actually everybody gets on extremely well. And for a school of pharmacy, that's essential, because we are essentially collaborative in nature. And also to develop the sort of medicines we're talking about here, it's also essential. So depending on what you believe about university ranking tables, and I don't blame you if you treat them with a certain amount of cynicism, At the moment in the QS rankings, UCL is ranked 8th in the world. And then a bit more information. So you wouldn't believe that this is the centre of London, but it's actually a very attractive central campus, which is very conveniently located near to King's Cross and Euston. So it's right in the heart of London, which is another big advantage to it. And there are many ways in which you can measure the success. So Nobel Prize winners have been mentioned a few times already today. This is John O'Keefe, who got the Nobel Prize in 2014 for his work on understanding how the brain can work out location. And it's a large university. It's a slightly chaotic university, if I'm absolutely honest, because it's grown by forming partnerships with institutions such as my own, which joined in 2012, and actually expanding that way. So it's actually more like a collection of villages. I'm going to that village metaphor yet again. But that's actually one of the strengths, because they work on the basis of get good people in and then support them and let them get on with doing what they do best, which is actually quite a good basis to form a university. What's quite interesting as well is if you look at the really big hitters in the UK, Oxford, Cambridge, Imperial, none of them have a pharmacy school. So we're actually in quite a privileged position of being a pharmacy school in, if you like, a super strong university. Of course, there are other very strong universities like Manchester, Nottingham and King's who do indeed have pharmacy schools. So my own institution was founded about 175 years ago as the education wing of the Royal Pharmaceutical Society, and then it became part of the University of London in 1926, and then in 2012 became part of University College London. And it's currently rated 7th in the world for pharmacy and pharmacology. If I was going to be very pedantic and a little bit mischievous about this, I would point out that many ahead of us, again, do not have pharmacy schools. They only have pharmacology schools, places like Harvard, Oxford and Cambridge. So we rank quite well as far as that's concerned. So it's a good relationship and we've integrated in very well. Now, you might think, is this all going to be about UCL? It's not. I'm swinging this gently into what I'm meant to be talking about. In terms of the current activities and priorities, I would point out two things from this list. First is the relationship we have with New Giza University. And Professor Farid, I think, gave an excellent introduction to this institution. And for me, what's wonderful about collaborations is when you transit from business arrangement to partnership through to friendship. And I think this is really what we've done, and we're working very closely with the team under the excellent leadership of Professor Manal to really try to instill in New Giza University Pharmacy School the values, the information and the possibilities that our students enjoy at UCL. And so far this has been going very well. But one of the points I'm going to be making later is that no matter how good your infrastructure and your strategy is, you've got to have suitably trained people and people with intelligent and creative ideas that you encourage in order to get to where you need to be in this particular sector and to be internationally competitive. And it was great hearing Medhat talking earlier on about the ambitions that they've got at NGU for actually entering scientifically into this sector. We also have quite a good history in terms of enterprise activities and kind of per capita of staff within UCL. I think we're one of the top divisions in the whole university for enterprise activities. So, to move on to ATMPs, and I'll focus more on them for now, but I'll swing back a little bit into things like super generics at the end. I think we've already had an excellent introduction into some of the therapeutic possibilities that are now emerging, so I'll deal with this quite quickly. So we have gene therapy medicines, and these, as has already been described, are really enabling us to insert genetic material or to modify genetic expression for a range of conditions. Cell therapy medicines as well are becoming huge as potential products, and tissue engineered medicines as well. And very broadly speaking, these are the three main areas that are considered under this umbrella term of ATMPs. And again, this has already been discussed by colleagues, so I won't spend very long on this. But if you look at the rise in this sector, it really is quite remarkable over the last few years. And the key thing here, of course, is that if something can be made to pay, then people will do it. And the converse is also true. If it's a nice idea, but basically at the end of the day, it's not going to be commercially viable to produce that medication, then it will flounder. But as we're seeing time and time again, and as colleagues have already mentioned, there are many products that are coming onto the market. But also another interesting measure, which I'll be referring to a few times, of activity, is looking at how many clinical trials are going on. Because it takes so long to get molecules onto the market or therapies onto the market, just looking at what's on the market is not necessarily telling you what's coming. Whereas if something has got into a phase 2 or 3 clinical trial, then the chances of it going onto the market are obviously much higher. And you can see there that there are huge numbers of clinical trials ongoing at the moment. So there's no question at all that this is a big growth area. If you look at numbers as well, then cell therapy is actually slightly more active than gene therapies and tissue engineerings, but this type of ratio could very easily change as time goes on. One of the comments, again, that has already been made, of course, is that we are looking for the foreseeable future anyway at low volume, high cost products. And so there are considerations as well about who is actually going to be able to benefit from these medications, how are governments going to cope with the costs associated with treating a single patient. But there are many mechanisms involved in that, such as treatments for rare diseases, from which we can actually learn a lot about the way that the human body works over and above alleviating the suffering caused by that condition. If you look at the distribution of where the clinical trials and activities and the startups are, then it's at the moment it's kind of concentrated in England and particularly the south of England. If you've been following British politics at all, which you probably haven't out of a sense of despair and confusion, which is kind of how we feel about it all, you'll be aware of the fact that there's actually a big emphasis on redistributing things away from London more in the UK, particularly towards the north. So that may change as we go through. But if you look at the sorts of things that people are working on in this area, most of it is actually in the oncology area, 60% of the clinical trials. There's also a lot on cardiovascular disorders, and there's also a significant proportion on central nervous system diseases. But oncology is actually a very big area as far as this is concerned, and indeed represents one of the areas whereby there have been some notable successes. Neurological diseases have always been and will always be difficult to treat. There's no easy solution as far as that's concerned. But having said that, the amount of research that's going on in the neurological area is absolutely huge. So again, these figures may change as we go through. And again, if you look at some of the cell and gene therapies that have been recommended by NICE, which is essentially the body in the UK that gives permission for medicines to be put onto the NHS, you can get some idea from flicking through this list. I won't go through them one by one, of the sort of areas that people are looking at. And as you can see, it's quite a wide range of both conditions and approaches. So a great deal of activity in this area. In terms of UCL, UCL is actually in quite an unusual position in terms of, again, if you look at clinical trials as a marker of activity, what you can actually, oh, sorry, I didn't mean to do that at all. Right, there we go, sorry about that. UCL actually has a disproportionate activity and presence in the clinical trials of ATMPs within the UK. The staggering, something like 59% of all such clinical trials are actually involving UCL at the moment. And I'll tell you in a minute why that is when I come to talk about support. The UK as well, you probably can't see the writing here, is very active in this area. Obviously, there are pinpoints in places like the States, places like Harvard and Philadelphia, where there is a huge amount of activity in and of itself. But what's quite interesting, if you look at the red here, the red is actually the proportion of clinical trial activity undertaken by UCL within the UK. And as you can see, it is a very significant percentage. And the sorts of things that have been looked at is particularly gene-modified cell therapies for rare diseases, T-cell engineering, and gene therapies, particularly using AAVs, viral introduction of the genetic material. So in a sense, the interesting question now, and particularly if the Ministry of Health is thinking about, well, how do we support the infrastructure within Egypt so that the success that has been seen by Menopharm can actually be reproduced elsewhere within the sector and indeed by Menopharm itself. This is just our experience. I won't go through all of these. I want to go to the bottom one first, strong educational and collaborative culture, and reiterate this point that any infrastructure that you put in place is always going to be supporting individuals who are imaginative, driven, and essentially collaborative in nature, because you've got to have the ideas in the first place. That means you've got to have the education system and the support for encouraging people to express ideas and to think outside the box and look at new paradigms, not just do what the previous generation has done before, in order to create the opportunities that that infrastructure can support. So although I put it at the end of the list, in many respects it should have come at the beginning. And this is why I think many of us believe that the university environment is basically, it's a breeding ground for everything else that is going to happen in the future. Even if the ideas don't necessarily come from the university, the people that will drive those ideas will. We have a number of organizations within UCL. So the Translational Research Office, for example, is a subsection of UCL entirely dedicated to that valley of death that I mentioned before. And they have a number of mechanisms whereby if an academic has developed an idea to a point where they are showing good potential for it to go further, they will then actually provide further support and guidance. Because the other thing to bear in mind is that us academics are not terribly good at making products. We like to pretend we are, we're not, is the truth of the matter. We don't understand regulation, we don't understand commercialism particularly well as a sector. So this is the support that UCL recognised was going to be required. We also have UCL Business and UCL Consultancy, and also we've got the Francis Crick Institute, which has opened recently, which is a collaboration with King's College and various funding bodies such as the Wellcome Trust and the MRC, which is the largest single-roof biomedical think tank in Europe. And it's only opened a couple of years ago, but we're expecting great things to come out from that. So there's a lot around us that is actually going to help us take these ideas, not just in terms of supporting it from a business point of view, but also from a clinical point of view as well, which is very, very important. So again, I'm not going to go through this diagram in detail, but this gives you some idea of some of the ways in which we as academics are being supported. So there's internal funding that we can get, so things like the UCL Therapeutic Acceleration Sport Fund. And you can apply for about 100,000 UK pounds, that's about 10,000 Egyptian pounds, sorry, my maths deserted me there for a moment. And that's to get you to the next stage. And then there's also mechanisms internally. And it's really this preclinical development phase. So what the university really concentrates on is the basic and discovery phase. Then the preclinical development, there's a kind of handover and several options there. But what's interesting is the external funding that's available from the government. And basically, what's really emerged is a kind of partnership between the university and the government, whereby the university says, look, we'll look after the very early stages, we'll filter out the ideas that are never likely to make it, but can you support the ideas whereby the evidence for potential success is actually pretty good? So for example, this MRC, which is Medical Research Council, which is a UK government agency, the Developmental Pathway Funding Scheme, DPFS, when you apply to them, and I've had funding from them myself, you have to give a really, really good business plan and timeline on all your activities. It's not a pot of money that you go away, work on for 3 years, and then come back with a nature paper. It's a, what is your development plan, and we will support you in doing that. And that works very, very well. And as you'll see in a minute, many products have actually reached market through this particular mechanism. And if you look at the portfolio of investments, so there's 91 total projects since all this started, of which 48 are currently active. But the really interesting thing is looking at the success rates that you get associated with this. So 14 of those have successfully transitioned preclinical to clinical. 8 projects licensed, 8 projects not going any further. That's going to happen. That's the whole point about having an idea and taking a risk. But 12 projects have been spun out into 8 UCL companies. So if you look at the hit rate, it's actually pretty good. And I think all credit to UCL that they have shown willingness to take a risk as far as looking at these sorts of investments is concerned. Because the key thing is that once you start investing in this and you invest wisely, then you start to find that you get the financial returns in a reasonable period of time afterwards. So alongside of this, there's also the, if you like, the scientific support that I've alluded to before. So we've got very good capabilities for manufacture and production of cell and gene therapies. We have UCLH, UCL Hospital, just down the road. And again, we have access to patients and access to academic-friendly clinicians as well. We have UCL Partners, which is basically a network of clinicians stretching right across London. So basically, to get to the clinical stage and to get to the stage whereby we can start to get clinical evidence for our activities, we're in quite a good position. And just to give you 3 examples of what this can mean, so Orca Therapeutics, which I'll talk about in a minute, has been floated on NASDAQ for $225 million, Autolus $160 million, and Amira GT $75 million. So what I'm going to do now is just to give you some examples of specific activities that UCL and the School of Pharmacy have been engaged in to give you a flavour of some of the projects that are either looking very promising or have already reached the market. So I mentioned Orchard Therapeutics. So this was focusing on ADA, adenosine deaminase deficiency. Used to be nicknamed, I believe, boy in the bubble syndrome. Basically, it is a genetic disease whereby the immune system of the child is very, very significantly compromised from birth. Without treatment, it is unusual for the child to live beyond age 2. And you heard earlier about some of the principles of these therapies. So it involves a bone marrow harvest, an isolation of the relevant cells, and transduction of the relevant gene, and then reinfusion of these genetically modified cells back into the child. But staying with the business side of this slightly more than the hard science of it, what's remarkable about this is that in a trial that was conducted completed in 2016, there was 100% survival in 32 patients that were treated, which is remarkable. Also, what's interesting is since then, GSK have signed a strategic agreement to transfer their rare disease gene therapy portfolio to Orchard Therapeutics. So basically, this is a huge success story run by some of our absolutely top researchers in the Institute for Child Health. And similarly, I mentioned earlier that cancer is a major target condition for gene therapies and cell therapies as well, of course, as you're no doubt aware. So the idea here is that Autolus was founded on the idea of taking immune T cells from the person's blood, growing them up, and programming them to recognize and kill cancer cells. Now, obviously, this is a description of the field as a whole, but with Puel's method, he used a unique logic gate method to improve that recognition of the cells. So in 2014, it was supported by UCL Business, UCLB, as part of the UCL Innovation and Exercise, and by 2018, it had been listed on the NASDAQ stock market and raised $160 million. That is not a huge time period. So there's a difference, and again, I'm sure colleagues around the table here know far more about this than I do, but there's a huge difference in raising the money to actually take ideas forward and necessarily getting the product on the market. Raising the money can actually be quite rapid, even if final appearance on the market is slower. So I'll now talk a little bit about, just to finish off, some of the ideas and activities that are taking place within the School of Pharmacy. And part of the reason I'm doing this, I mean, I'm director of the School of Pharmacy, so naturally I'll come to meetings and go on about what we're doing within the school, but also because I just want to show what it's possible to do in a School of Pharmacy. I was delighted that Professor Farid's comments about the fact that schools of pharmacy are therefore so much more than to dispense medicines, important that that is. They're vital healthcare professionals, but they're also vital contributors to the scientific community, particularly in the areas that everybody in this room is particularly interested in. So this is why we are delighted to be supporting Newcastle University, but also hopefully supporting the education sector in other ways within the country if we can. So this is my colleague Stephanie Shorg, and we stole her from the Institute of Neurology down the road, which is one of the top neuroscience institutions in the world. And she works on epilepsy. Now, everybody knows that there are a number of drugs that can be used to treat epilepsy, and most patients have their epilepsy and their symptoms pretty well controlled. However, what is less well known is the fact that about 25 to 30% of epilepsy sufferers do not have their symptoms well controlled at all. And this is known as refractory epilepsy. And it's a life-threatening condition, and very often the only course of action open to them is major surgery, which of course is very undesirable indeed. And basically, you'd have thought, well, this is gene therapy, so this must be about a genetic defect, so it's only going to be those people born with this defect that this can help. But that's not actually true. Gene therapy can also be used to express proteins which will control or modify cellular behavior, even if it isn't necessarily replacing a faulty gene as such. And that's what this approach does. It basically codes for a protein which suppresses seizures. It suppresses the unwanted brain activity. And just to very briefly walk you through this diagram here, it's actually quite straightforward. So the black line here is the control of non-epileptic. The red line is the sham of empty AAV viruses. But what you see with the treated is that after a single treatment, you can see that the seizure-like events actually reduce down to a non-epileptic model. Now, this is a rat model, but the potential for this is hugely exciting. The implication is that this could, with one dose, potentially remove the seizures from a patient. So Stephanie's been awarded about £2 million from the MRC, again on one of these translational pathways, to actually develop the evidence to go into humans. So we're extremely excited about that. Ahad Rahim, again, another very valued colleague working in the gene therapy area. He works in rare diseases. And his approach, and I remember his interview, and our jaws were on the ground when he was presenting his work, and I think I was the one that said to him, is this actually possible, what you're proposing? He actually proposes to treat the unborn child. He basically proposes to treat the child in the fetus during pregnancy. And the models have indicated that this can indeed actually work, and there are significant advantages to doing so. One of the diseases that he works on is Gaucher disease, which is an extremely cruel genetic disease, whereby the lifespan is well below 3 years. And essentially, it's a deficiency in a gene expressing a vital protein. And so what he's been working on is using AAV-mediated transfer of the gene, but into the fetus. And what he's shown in a rat model is that this can actually significantly alleviate the manifestation of the disease in that model. And very recently, the venture capitalists have got hold of him. And I'm not allowed to disclose the sum, but again, it was another eye-watering sum that they're going to be investing in the technology that he's developed, because they believe that this can actually be brought onto the market. Also staying with ATMPs, but moving slightly onto regenerative medicine, James Phillips is working on peripheral nerve damage. And essentially what this means is that if somebody particularly has an accident, then over and above the damage, their blood vessels and their bones, you can also get severed nerves. And very often it's the severed nerves that cause the damage. That's what causes paralysis, for example. Now, the good news about nerves is that they do actually try to grow back together. The bad news is that if you've got a significant severance, then the chances of them finding each other, or the two ends finding each other, is pretty much zero, and the nerve just gives up and you end up with permanent paralysis. What James has been working on is two approaches. The one that is really sort of getting pulses racing is these tubes whereby you can actually insert the different ends of the nerves in either end in order to guide them through so that they actually find each other. And what's particularly interesting about this, and it goes back to the idea of multidisciplinarity, the person that he's working most closely with on this, yes, is a nerve specialist, but also is a mathematician, a really brilliant mathematician called Becky Shipley. And she is an expert on biological modeling. So she was able to supply the know-how for how exactly will you be able to construct this collagen matrix to get the different sides to work with each other. So again, they've just got a multi-million-pound venture capital investment to actually take this to market. We've also, in the low molecular weights, because I don't think it's true to say that low molecular weights have had their day, there's a lot of conditions whereby they have an incredibly important role and will continue to do so. But Stephen Needle has developed not just a sort of first-in-man trial, but a first-in-type trial, whereby he has developed molecules that attack what are called holiday junctions in the DNA. These are little glitches in the DNA, which have been found to be very specific for certain tumor cells. So actually, that gives you a way of differentiating between tumor DNA and healthy DNA. And pancreatic cancer, we have not showered ourselves in glory over this. The survival rates have not really improved much in the last 40 years. But things are looking very hopeful in the preclinical trial with this molecule, and hopefully it will go into humans. Matt Todd is a recent acquisition. He was in Sydney, well, he's an English guy. He works on a thing called open source drug discovery, which is like crowdfunding, but with intellectual input to make new molecules. So what happens is that people actually contribute to a website of finding cheaper and more effective ways of manufacturing new drugs or manufacturing existing drugs. The schoolboy that you see there, you may be familiar with the pyrimethamine story, whereby Schreckl increased the price by 5,000% on the extremely dubious basis that it was extremely difficult to manufacture. Using this approach, a bunch of Sydney school children manufactured exactly the same drug for about $1.90 per tablet, whereas Schreckl was actually trying to charge several $1,000s per tablet. So this is something we're very excited about, particularly in the treatment of diseases such as malaria. And then finally, just going to the super generics, this is Abdul Basit, and he's been working on a film coating system which will do absolutely nothing until the tablet reaches the colon. And then there's a unique dual mechanism whereby the tablet coat disintegrates, and it allows you to specifically target drugs down to the colon. And for things like Crohn's disease, which affects more than 3 million people in the United States, there is a huge need for this, because the way in which the tablets are taken currently is very inefficient. The patients have to take the standard 400 milligram tablets 3 or 4 times a day, and even then, when you look at the remission rates, they're not very satisfactory. With mesalazine, I think, is the drug that was used here, the remission rates for his novel formulation are much, much higher, and it's once daily dosing as opposed to multiple daily dosing. So this is very shortly going to reach the market as well, so we're extremely proud and pleased that Abdul has developed a system which is going to really make a difference to people's lives. So concluding comments, I think it would be unnecessary for me to really hammer home the point. Things are changing. I think every speaker this morning has made that point very, very clearly. And I think, all credit to the Ministry of Health and to the companies such as Minopharm in Egypt and indeed to NGU for embracing this and bringing together this meeting to look at ways in which we can do this. Don't forget the university sector. We can contribute an enormous amount. We can contribute ideas and early stage products, but we can also contribute the people that will go into the companies or go into the ministries in order to make this stuff happen. And it's all about partnerships. It's all about finding ways of understanding what each other can contribute, because the universities can't do it alone, but then no single sector can really do it alone. And I think you heard from the Minopharm story that really it was a very effective series of partnerships and good strategic thinking that actually led to these successes. So thank you very much indeed for your attention, and I look forward to listening to the rest of the conference. Thank you.
Okay, thank you very much for that very valuable presentation, and now we can take a break for I think 13 minutes and then rejoin to this conference.